Hello, everyone. It's 1 p.m. here in Bethesda, so let's get started. I'm Sheetal Jose, and I'm a program analyst at NHGRI. I would like to welcome everyone to this pre-application webinar for the eMERGE Genomic Risk Assessment and Management Network RFAs. Before we get started, I wanted to go over a few instructions for the WebEx. So this webinar is being recorded, and it will be available for viewing on the website. I have posted a link to the website on the chat box. Please note that participants will be muted during the presentation, so please hold your questions till after the presentation.
Hello, this is Rob Rowley. I'm with NHGRI. I'm a program director with eMERGE, and we will be talking today about the Genomic Risk Assessment and Management RFA. So to start, I'm going to go through a quick overview of the talk today. First of all, we're going to talk about the Genomic Risk Assessment and Management overview, which will include first key definitions. We will then talk about the purpose of the RFA. We'll then talk about the objectives of the RFA. Then there'll be a specific section on the informatics, since it's a significant portion of this RFA. We also talk about NIH clinical trial policy and why this is a clinical trial. Then there's some specific information about clinical sites that we'll touch on. Then we'll touch on the specific issues related to the coordinating center. We'll talk about the FAQs that we've received after the FAQs were originally published, and then we'll open up to applicant questions. So to kind of jump into this, we'll talk about key definitions. If you look at the key definitions, one of the things that comes up often is what is a genomic risk assessment versus a polygenic risk score. The thing that's critical to understand is that the genomic risk assessment is a calculation of risk based on, can include a polygenic risk score, but it also includes family history and other clinical information to make that calculation. So talking about a polygenic risk score in terms of a risk assessment tool, it actually looks only at the allele frequencies. So again, genomic risk assessment can include polygenic risk score as part of that. Family history for this RFA is defined as patient-driven. It can be web-based tool that integrates with EMR, such as Ignite's MeTree. There have been questions related to this and whether that you could use other tools. MeTree is not defined as the only tool that can be used, but anyone that can capture a family history. Clinical information includes non-genomic information such as lab values, anthropometric values, habits, and past medical history. And then really talk about risk management. This is the part is once a risk has been assessed in a patient is to really determine what further testing needs to be done as a result of that risk. And this could include both testing as a bone density or mammography, or it can include interventions such as drug treatment, surgical removal to identify and treat to prevent the early disease that was identified with the risk score. We talk about clinical risk estimation. Again, this is the result of a genomic risk assessment, is what is that value or what is that risk of that individual based on all those findings at the end of that evaluation. So I'm not going to go through all this, but there's often questions about diverse ancestry, and this includes non-European plus Hispanic, Latino ethnicity, and there is a directive that we've outlined there that you can, and we'll provide the link for that in the RFA. This includes American Indians or Alaskan Natives, includes Asians, Black or African Americans, Hispanic or Latino, including Native Hawaiian or Pacific Islanders. To talk about underserved populations, there's actually a publication for that, and we will refer you to that to get further information. So what we're going to do now is kind of look at what the purpose of this RFA. When you look at the purpose of the RFA, we really have broken it into two areas. The first here is really to kind of understand and calculate validated polygenic risk scores for several complex diseases, and we define 20, defining 5 in detail and 15 just by list. And what this will include is kind of defining what data sets that will be needed and how are those risks calculated on that polygenic risk score. And then the other part of this is that questions have arose is how can you do this in populations that haven't been studied for? This first year will actually be spent determining how to calculate those risks in those populations, and we're looking for proposals on how to do so. The next thing is, as this is going on, the sites will have to figure out how to implement these electronic medical record-based methods to communicate that genomic risk to the different individuals. And in addition to this, we'll need to figure out how they're going to capture the necessary data to calculate a genomic risk assessment. Once this is determined, the idea is to recruit and genotype 20,000 individuals of diverse ancestry and then prospectively calculate these risk assessments and provide those to the clinicians taking care of those issues as it relates to their primary prevention of disease, and then follow them longitudinally, and from that, use the electronic medical-based records to assess what outcome and the uptake of the risk reduction recommendations as we go forward. In terms of the objective of the RFA, the objective is, again, looking at retrospectively, we'll need to validate these different tools. And again, we'll describe 5 in detail and 15 potential for inclusion. We'll identify the appropriate thresholds. So what threshold will be adequate to determine that they're high risk, and this will need to be defined. One of the questions that's listed there is guidelines. This can also be expert opinion in terms of how individuals want to establish that risk level. We'll also need to determine the distribution of risk across the key demographic subgroups, and then we'll need to modify that accordingly. We'll compare these risk predictions with specific e-phenotypes. We'll modify those thresholds or phenotypes as appropriate, and then really determine the feasibility of the added value of genomic information to these risk estimates, and then base it on both clinical and non-genomic information in addition to the alleles. So this, again, for the second phase is really looking at the objective from a prospective risk assessment. The idea is to recruit 20,000, approximately 2,500 per site of diverse ancestry to undergo genotyping and determine that genomic risk assessment. And again, this can include a variant assay, but also include family history and clinical evaluation risk to determine that risk. What we'll do is the steering committee will actually come up with a list of 15 risk estimates and then implement those across the population of 20,000. And then we'll arrange for follow-up testing and risk management of the patients who exceed agreed upon threshold as needed. And another thing that will be needed is how to monitor these individuals. The idea is that if we take this approach, if we don't have an uptake of approximately 50%, the steering committee and the group investigators will need to determine if they need to continue that or modify the protocol based on that. We'll need to be able to disseminate and analyze this rich data set. In order to complete this clinical trial, you will need to capture data from all the sites. This will include clinical genetic information, and this will be analyzed centrally to be able to understand what the impact of each of these risk assessments are on the patient population. So risk management, this can include interventions tailored to individual patients' estimated risk. The risk management recommendations delivered to the patients exceeding an agreed upon level. We'll design components of the genomic risk assessment, again, to include risk reduction recommendations, and this should be proposed by each applicant. Again, reinforcing the single network-wide protocol for the risk assessment and risk management and outcome ascertainment will be finalized by the steering committee by consensus. The risk assessment and management will be updated again, depending on the uptake if it drops below 50%. And again, this will be finalized by the steering committee by consensus. The idea in terms of collection of outcomes, understanding this is over a 4-year period, we'll have to be mindful of that in terms of when we're looking for outcomes. So this can include hypertension, obesity, health behaviors that can be extracted from the electronic medical record. As we're following these individuals, we'll look at those outcomes. It can be such things as disease surveillance or screening consistent with guidelines. It also can include if individuals that meet certain criteria that the drug is selected and consistent with guidelines or expert opinion. And then again, the question will be, will there be improvement in modifiable risk factors, for instance, cholesterol or blood pressure after the directed care? Other guideline-directed care, such as drug treatments and surveillance practices, will need to be followed also. And then another component of this can be cost and utilization of healthcare in these high-risk groups. Again, getting into the informatics.
Hi, my name is Ken Wally, and I'll give a brief overview of the informatics for the RFAs for eMERGE. Go to the next slide. So, informatics has always played a key component in eMERGE. and its utilization to reduce barriers for advancing genomic medicine. For these RFAs, what you can expect informatics to actually be utilized to actually integrate family history, genetic testing results, polygenic risk scores, and clinical decision support into local electronic clinical workflows. We look at informatics as more of a comprehensive approach to try to address these barriers. However, we don't want things to go in off the rails or in different directions, so we want to actually leverage existing fast-tail interoperability resources standards for our ability to transmit and develop utilization standards for genomics and clinical practice. In addition, we want to have a central place to provide network-wide analysis using the NHGRI Data Science Analysis Visualization Informatics lab space. Additional information that you should be considering in your applications is that the eMERGE network leverages the Observational Medical Outcomes Partnership data model. For ePhenotypes that were briefly discussed, we'll also include expanding those efforts to address both the structured and unstructured components by leveraging natural language processing. So your application should reflect expertise in that area. Next slide, please. For those who are not familiar with the NHGRI Analysis Visualization Informatics Lab Space, they also call this the AMVL. The AMVL is NHGRI's cloud-based resource that provides an opportunity to do shared analysis and computing. We'll also have both open and controlled access data. It's also going to provide cost control for cloud services and user training for this resource. This cloud resource is not developed in silos. It's going to be part of what we call a federated data commons ecosystem with the ability to try to work with other commons such as those established by other institutes so that individuals will be able to access data sets and do their analysis. For cost control, users should expect to for data sets that are being hosted to be made available to the public. The AMVL will provide funds to cover the cost for those data sets. However, all other data sets for analysis will need to be covered in the users. So make sure that your application reflects the budget for the use of this resource.
So now we'll talk about the NIH clinical trial policy. And some of the questions that have come up is, why does this meet the NIH definition of a clinical trial? In 2015, the NIH expanded its definition of a clinical trial, and the criteria for that included it involves human subjects, it evaluates the effect of the intervention, it monitors the biomedical or behavioral outcomes, and it prospectively assigns to an intervention, which is all consistent with what we're proposing here. But we are not expecting a traditional randomized clinical trial for this RFA, but we are accepting small pilot studies that are proposed within the individual applicant. And this is a reference to that. So one of the significance of being a clinical trial, I'm not going to go through all these, but I'll kind of highlight the different bullets. First of all, the applications is evaluated based on their specific criteria. So look in the review section. Those criteria are a template for all clinical trials. Another part of this is the network must register with clinicaltrials.gov, and you can see the requirements underneath those under those bullets. The other part of this that impacts investigators is they do need to require good clinical practice training for all investigators, and that's defined on the link that's listed there. The other part of this is principal investigators must submit human subjects in clinical trial information form. And again, this is a delayed onset human subject. It's not a delayed start submission. Principal investigators must submit updates using the human subject system that's available. And principal investigators must post clinical trial informed consent after the completion, at least 60 days after the completion of the IRB approved consent and the end of the trial. The network must submit a single IRB submission, which will be coordinated across the network.
Hi, everyone. This is Nicole Lockhart. I'm a program officer in the Ethical, Legal, and Social Implications Research Program, which is part of NHGRI. This RFA also features a small LC research component. Applicants are encouraged to propose a small 2-year LC research study. That study should focus on unique LC research questions raised by the calculation, return, understanding, use, and utility of polygenic risk scores in diverse populations. The study could utilize empirical, legal, and/or normative methods as appropriate to the study design. And the LC study should be integrated into the larger eMERGE protocol that the applicant is proposing, such that the LC research informs the subsequent implementation of polygenic risk scores within eMERGE.
So getting into some specifics in terms of the clinical site, first of all, you're able to recruit and follow 2,500 patients in a clinical setting who have not previously received genomic information. There are some questions on whether that if a patient has certain testing, germline testing, can you use a polygenic risk score to further classify that individual? If the individual proposes that, they would have to explain how they would leverage that across the entire network. The other thing is able to perform a genomic risk assessment, including polygenic risk scores and the appropriate clinical management for those patients. Able to implement family history, genetic testing results, clinical decision support, electronic clinical decision support, and also perform the outcome assessment in a comprehensive electronic medical record. And one of the things that's critical for this work is the standard is to collaborate and be able to express and share ideas across the network. In terms of talking about clinical budget assumptions for this, there's 8 clinical sites recruiting 2,500 patients each. Each clinical site will need local IT support or informatics support, and will need to estimate cost to integrate the family history, clinical decision support, and the testing results into the clinical workflow. Each site will need to estimate the cost to capture and transmit the data to the central repository for a network-wide analysis. Other things that should be taken into consideration is each site should include a patient-related clinical research cost not covered as part of the patient's existing health coverage. For the Enhanced Diversity Clinical Site RFA, it's estimated $1,100 total cost over the entire duration of the study. For the clinical sites, it's estimated for $900 per high-risk individual for the duration of the study. Each site will need to estimate the necessary personnel and resources to return and manage these genomic risk assessments. In other words, is once you calculate a genomic risk assessment, individuals will have to return that information and be responsible for managing those individuals throughout the course of the study. And we'll go into the coordinating center, some specifics there. Again, it's expected that the coordinating center will be responsible for subcontracting and network-wide genotyping of all the participants. It'll need to be CLIA and FDA-approved SNP genotyping assay. They'll oversee the network-wide deposit of data into the Anvil or a central repository. Again, they'll need to ensure that the network has the necessary tools to analyze the network-wide data set. They'll need to coordinate with the network, NESP, periodic data reviews, and they'll oversee the single IRB submission and also registration with clinicaltrials.gov. They'll also need to be able to ensure the monitoring and variant interpretation and up-to-date genomic risk assessments in case these change over time, depending on reclassification. In addition to this, they'll be responsible for ensuring that the network keeps up-to-date management tools. And again, the coordinating center, as they currently are, is responsible for paying and coordinating all network-wide meetings in conjunction with the steering committee and NHGRI. Now we'll go into the FAQs.
Hi, my name is Jill Leal. I'm a program director for eMERGE. We published some FAQs and we've received some questions since then. So the following FAQs is our response to those questions. Okay, are sites required to have both pediatric and adult patients? Yes, applicants may choose to focus on adult or pediatric conditions based on their local expertise and interests, but should demonstrate their ability to develop and manage both pediatric and adult conditions. Will sites be required to implement all 15 genomic risk assessments? Yes, genomic risk assessments are expected to be implemented for primary prevention for all patients. Applicants will need local expertise to create the genomic risk assessment, but will be able to use the expertise across the network when implementing. Can patients who have had genetic testing be included? Yes, patients can be included if they have had genetic testing, as long as they have not been returned to the patient or their provider. The RFA indicates that the clinical site will be expected to return secondary findings not included in the reported polygenic risk score. What are secondary findings as it relates to the RFA? Secondary findings are genetic test results that are not part of the primary purpose for performing the selected genomic risk assessment.
What data will be required to be deposited into the central repositories like the AMBL? Clinical history and examination data that includes family history, laboratory findings, and imaging as relevant. Also, genomic data as well as calculated clinical and genomic risk scores. All delinked from personally identifiable information will be deposited into the central repository for network analysis. What is Fast Healthcare Interoperable Resource, FHIR, and how are we planning on using it for the eMERGE network? So FHIR is an HL7 interoperable specification for healthcare interoperability. eMERGE plans on leveraging FHIR to capture and return structured genotyping and clinical reporting of health information. We understand that there are some limitations to FHIR, but we also want to utilize FHIR and expand on it for the use for eMERGE. So this is in terms of acknowledging all the people that have contributed to the previous phases of eMERGE. I always like to use that there's 136,000 patients that have agreed to have their results used for the eMERGE network. And kind of to put it in perspective, it's the city of Savannah. So when I look at I thought that was kind of interesting that that's the number of people that have participated in eMERGE so far, and we plan to continue that. The other people, as you can see, there's multiple groups involved with this effort, and it's been ongoing. And the collaborative effort here is quite amazing. So just important dates to think about. The open date, which is the earliest submission, is July 2nd, 2019. There is a letter of intent due date, July 2nd. Again, this is optional. And the application due date is August 2nd, 2019 by 5 p.m. local time of your organization.
I have a question. My apologies if you've already brought this up, but can you remind me who is doing the reporting for the polygenic risk scores? Does that come from the genotyping lab, the coordinating center, or the individual sites?
So when you look at, I guess if you look at the polygenic risk score, I would combine that into the genomic risk assessment. The expectation would be that clinicians that normally are taking care of these patients would return those results to the patient or participant.
Yes, I understand about returning results, but like in eMERGE 3, the genomic centers are generating the genetic report based on the genetic results. Am I assuming there will be a similar type of report from the genotyping center for eMERGE 4 that will be then ingested by the individual sites for the overall interpretation?
No, so this is Chairman Olio. So that's something that's going to need to be developed by the steering committee. So we'll have to figure out as a group what we will do with the genotyping information from the genotyping center that will come through the coordinating center. But all of that will need to be developed by the steering committee, including what clinical decision support is provided back to the clinician and the patient. Does that answer your question?
Yes, thank you.
So we have a few questions on the chat box.
Okay.
So
I can take the first one. So the first question is, if there is a single protocol that has not been defined, how are we supposed to describe how we will integrate this with our EHR? Currently, EHRs are allowing extremely limited inbound FHIR connections or required to support inbound FHIR. That's a very good question. So for this phase of eMERGE, as I mentioned earlier, there are things, we understand some of the limitations associated with FHIR, but we also want to leverage FHIR and expand on that. So expand on those efforts that are already been developed. eMERGE is already in the process of developing one FHIR standard for reporting structured sequencing results. For this, to answer this question, we actually want you to think about how you would leverage FHIR in your application. Understanding that there needs to be flexibility for eMERGE as a group to actually develop processes to actually address some of the barriers that we are going to face with the FHIR implementation component.
Does that answer your question?
It answers the FHIR part. I guess the issue is when it comes to returning results and collecting data and exactly which scores are going to which specialties and things like that, that can vary a lot based on local implementation of even the same vendor EHR. So without any of that detail, what are we supposed to be describing?
So you're actually describing, one, your commitment to leverage FHIR. You're describing how you plan on leveraging FHIR in your analysis. What we're trying to do is provide some flexibility for you to give us your ideas on how you would utilize this resource. And identify those barriers that are associated with that, with trying to implement that, using FHIR in that process. So this is something that we're really looking towards the applicant to help us to inform best practices for leveraging this resource to address those questions.
Yeah, so I think then just to expand on that a little bit, obviously, you know, we propose many different technologies and approaches at the beginning of a program that are developed during the course of that program. And typically it's best for applicants to describe how they would see that being implemented within their system, whatever the best thing that they can propose and identify. But then express their willingness and demonstrate their ability to adopt what is agreed upon by the steering committee.
So does that imply that our alerting and results presentation and those sorts of functions are supposed to use FHIR-based tools rather than native decision support in our EHR?
Well, you also have to factor in where you are in your development and implementation of FHIR. So one thing would be that you may not be able to take it all the way end to end, but there may be a process that you could. You need to itemize, you need to acknowledge that in your application and also show that you're even willing to test some of these other FHIR resources in your system. What we're really looking for is people who and applicants who have an idea on how they want to leverage FHIR and also willing to work to adapt their systems to utilize FHIR. Understand that those barriers exist and that we're going to have to figure out a lot of these processes as far as best practices to implement this.
Okay, thank you.
So we had another question here. Does return genetic testing include consumer testing such as 23andMe? The challenge with that is going to be if it's included within the genomic risk assessment, yes, it would. Okay. Any other questions? If not, we have one more. Confirming that the clinical sites are not responsible for the cost of the genetic test. That is correct.
That's for the network-wide test, but you need to, there may be follow-up testing.
Yes, we have to be covered by the clinical sites. But that would be part of, too, is expecting that might be part of routine care and might be covered by their insurance.
Right. So it's the network-wide testing and only that we're anticipating. Things can change, but what we're anticipating is that would be paid by the coordinating center. And then if there's individual follow-up needed at a specific locus or whatever, that would be something that the clinical sites would cover for their patients.
Okay. Here's another question. What year do we expect prospective recruitment to begin? It'll be one year after award.
And on the next question, can you please confirm that it is a requirement to enroll both adults and pediatrics?
Yes, it will. So in terms of proposal, it's based on your local expertise to actually develop the polygenic risk score. We'd expect you, or the genomic risk assessment, we'd expect you to propose that based on your local expertise. But once the cohort is engaged, it would be expected that these These 15, if they include a pediatric, they would include both adults and pediatric patients at that time.
Right, so the clinical sites will want to demonstrate their ability to enroll both groups of patients. You might also look at the current distribution and anticipated distribution of polygenic risk scores that might be reported back, and ask yourselves, are these more likely to be in adults or in children, and perhaps structure your application accordingly.
I had a basic question about the recruitment strategy. If the goal is to recruit folks with high polygenic risk scores, can you leverage existing data about polygenic risk scores to re-recruit them for clinical genetic testing?
Just 1 minute.
So, can you hear me?
Yes.
Okay. Yes, the goal is not to recruit people with high polygenic risk scores. The goal is to recruit just the average person, and then what we would do is calculate their genomic risk assessment and polygenic risk score and determine that they're high risk based on that.
Right, so the expectation across a large group of diseases is that many people are going to be at high risk for something. What we're estimating is if we take the top 2% of risk, that 25% of the population will be at high risk for at least one of 15 diseases. But the goal is not to preferentially recruit people at high risk. Does that make sense?
Got it. Thank you.
I'd like to take one of the questions on that we received was how should we budget for egress and computing on the Anvil? Oh, sorry. One of the questions that was submitted was how should we budget for egress and computing on the Anvil? There's the link. The Anvil is built on top of the Google Cloud platform, and so we can, if you go to the website for either the Anvil or for the eMERGE RFAs that was shown, you can actually see a link to where you can do pricing figure out pricing for the egress and computing on the Anvil. I want to be clear that the data sets that are deposited into the Anvil that are available for the public to access, either through open or controlled access, those data sets would be covered, storage costs would be covered by the Anvil. However, all other data sets will be required to be covered by the users. So please also include the cost for storage in your budget, along with your egress and computing requirements. The next question is how to analyze information. And I was curious if who submitted that they could elaborate what they mean by that question. I guess not. So I'd like to go to the next question. What approach do we use to analyze the genomic information?
That was a question.
That was the question.
Oh, how to analyze the genomic information. So sorry.
Okay.
So the next one, given develop and deploy polygenic risk scores for minorities, can you provide the minority representation for the 136K eMERGE participants? So this information is actually, I believe this is on our website, is that correct? Yeah, so we can send you the link for the eMERGE website that you can look for that information. The next question, will these be developed for minorities in year 1?
I think the key word is not necessarily developed, but adapted would say that existing polygenic risk scores, how would those adapted and be validated in those populations?
So the plan is to start with existing polygenic risk scores, but understand that we anticipate that these risk scores would have to be modified. They'd have to also be developed. New ones have to be developed to address the broad diversity of the population we're trying to recruit. So please keep that in mind as you're planning your application. Also, finally, can you add data from minority patient cohorts to enable more robust PRS scores? Will you answer that? Yeah. I was going to say yes. Yes.
So there is a question about limiting analyses to the 83,000 existing eMERGE participants.
Yeah.
Okay, and that's not what the RFA says. Can we expand on that a little bit?
Sure. So any publicly or any data set can be used for this analysis. It just needs to be described and in their proposal.
Yeah, we just mentioned that as one potential source, but that's obviously not the only potential source.
So next one is the plan to have a central consent form which will be used for all clinical sites?
Under the single IRB, as you know, the informed consent might vary slightly from site to site, but the idea is to create a single consent form.
So yes, that's what we're aiming for.
As the network will identify high-risk patients, is there a plan to recruit families in the future or in this RFA?
There's nothing excluding recruiting families, if that's but there's no plan to recruit specifically.
And applicants can propose whatever they think is the best approach to that.
Right. The budget as described for the clinical sites does not cover costs for CLIA blood draws and DNA extraction for the 2,500 patients. Is that included in the $900 to $1,100 per patient cost?
No.
Right, so that will have to be covered, as part of the clinical site budget.
Okay. Are there e-phenotyping protocols already developed? Those research developed methods for e-phenotyping, or would this be unresponsive to the RFA? Yes, we do have electronic phenotyping protocols. eMERGE has a long history of developing these protocols and algorithms. And yes, we would like to, you are encouraged to propose new methods for e-phenotyping. As mentioned in the presentation, eMERGE will propose to leverage both the unstructured and structured components of the EHR to actually help improve our efforts for developing e-phenotyping. And we look to leverage NLP processes to help perform those efforts. In addition, eMERGE also leverages OMOP for our data model, and that should be reflected in your application if you're interested in advancing or producing e-phenotypes.
Next question is, you said 15 phenotypes polygenic risk scores will be implemented in the EHR and returned to all patients in primary care setting. When and how will these 15 phenotypes be selected? Should our recruitment ensure the prevalence of these 15 phenotypes in our cohort? Again, during the first year, you should propose what your local expertise has, but in the first year, the steering committee will decide on what those 15 phenotypes are.
So it's the goal for the polygenic risk score to be a feature of a general multivariate predictive model that includes other clinical and demographic features. Or as intended, we will evaluate PRS for predictive properties alone. Since part of the goal of eMERGE is to actually understand more about what's needed to develop polygenic risk scores for diverse populations, we're having a broad approach in this. So your application should reflect what you believe is the best approach in actually addressing those polygenic risk scores. So that should be reflected in your application.
Can you clarify the classes of data needed to be abstracted from site EHRs? Does imaging data include reports and metadata, or also the images themselves? This will need to be determined by the steering committee based on what genomic risk assessments are decided by the steering committee, and also include the management recommendations that the network decides on will determine the results of that.
We received a question. What sorts of LC research do you anticipate, particularly since it will be done before the testing has been done? I'll take that, and then we can move to the second half of that question. So we anticipate that this would be exploratory research, kind of modeled around something like an R21 in terms of scope and size, to take place the first 2 years of the award. So this would be something that is tackling unique LC issues related to polygenic risk scores. I think that could go in a lot of different directions. It could look at normative issues. It could look at patient understanding or perception of polygenic risk scores. You might want to think about what LC issues you would want to study before implementation at your clinical site. So I think we don't have a specific project in mind. We're really looking to applicants to try and identify what they think are the most pressing issues there. The second half of that question, which I will let someone else answer, is do you anticipate limiting return of secondary findings to the ACMG 59?
Yes, so it is expected that if there's any findings that are found on the testing that are not included in the genomic risk assessment, those will be returned to the participants.
Right, but the selection of those would be whatever the current standard is, and as the questioner pointed out, that the current standard seems to be the ACMG 59, but we would expect the steering committee to discuss that and come up with either an evolving standard or some other agreement as to what should be returned.
Do we need to include a detailed analytic plan and power calculations? Yes.
The website, anyway. I wanted to tell you about a conversation that I...
Yeah, Scott, you are muted. You may want to mute yourself. Good. In terms of the, obviously, you need to structure your application. You don't have a lot of space, so you need to structure it in the best way you can to meet the review criteria, and that would seem to be one of the components you need to include.
And that and you can also consider you will have the clinical trials pages where you can explain your design, so some elements may fit there.
So given the requirements for providing risk estimates to patients through ECDS incorporating the EMR, and the requirements for online tools for patient-centered family history, does this require integration with patient portals? So though it's not a mandatory component, we are encouraging applicants to show an end-to-end process for disseminating and return results. And we understand that there's going to be variations in how that is done, but applicants should propose how they would do from end-to-end return results to patients and collecting that information, whether it's using patient portals or other venues. But we would like to see that more reflected in your application. We encourage the use of informatics to provide that resource, but we will leave that up to the individuals to decide what's the best route to actually disseminate that information.
Next question, what would you say the major activities of the local experts in year one would be? Will these activities include local data or data available in eMERGE? The data, it depends in terms of what data is available from eMERGE at the time, but the hope is there's 83,000 samples that are being deposited in dbGaP at this time that should be available at that time. The local experts in year one would be the individuals, as you're going to propose different genomic risk assessments for those patients. That's your local expertise, and the expectation would that you'd bring that expertise to the steering committee and help shape and form in terms of what are the cutoffs for risk and what would be the management plans for those individuals.
Next.
Oh, sorry. You note that the network will use GRE rather than TRS. Since GRE would be novel and would derive from the network, how will these be externally validated prior to returning to patients? A very good question. So this would be something that the steering committee would actually put together to address, but we encourage you to submit a response to how you would approach this.
I think I see some others that haven't been addressed. Should we propose 15 or 20 conditions for genomic risk assessment? We know that that's a confusing issue. So someone asked, as part of the application, should we propose 15 or 20 conditions for genomic risk assessment? That is, are we proposing a complete network study along the lines of the 20,000 recruited patients? So if you look at the RFA carefully, what we were initially suggesting, and again, all of these plans need to be discussed and finalized once we have awardees and put a steering committee in place. But the idea is to have a larger set of scores that we would then choose among in order to have the subgroup that will actually be implemented in patients. And there are a variety of criteria on which you may want to choose among those, and the steering committee will decide on those as well. For the purposes of the application, we're asking each applicant to describe 20 polygenic or conditions with polygenic risk, with genomic risk estimations that they would see as being appropriate to be considered. We recognize that if we have 8 or 10 applicants, there's going to be more than 20 that everyone will propose, and so from that we'll bring down to the 15. But note that in the application, since you only have limited space, we're suggesting that you take 5 of those conditions that you feel most strongly about or have the most expertise in and describe those in much more detail, and then the other 15 you could just sort of list that those would be others that you'd consider. That's just sort of to give you and the reviewers a chance to be able to get all of this done in the time available.
So who will generate the PRS for applications in the site? Will the supporting center or the contractor do this, or is the CC responsible for deciding what PRSs to apply in their settings? In your application, you should include PRSs that you are interested in developing. However, understand that the steering committee will work also to have a cohesive group of PRSs that the groups will also take.
Next question, what is the time frame to complete recruitment? The expectation is that the recruitment will start at the end of the first year and continue through year 4. The next one is, the sites will extract DNA and then transfer. Any other sample? As mentioned in the RFA, if there is genetic testing that's network-wide, that will be coordinated that way. Otherwise, it will be standard of care in terms of how those samples are managed.
In previous eMERGE projects, subgroup projects were encouraged. Our sub-projects allowed or is meant to be a monolithic network, single project where opportunity to do sub-projects is discouraged. That is not the history of eMERGE. It's not considered a monolithic, well, at least my perspective is not a monolithic entity like that. eMERGE is really leverages the ability to bring diverse ideas and concepts and work together to try to solve them. So subgroups are encouraged, so you should feel free to submit ideas for sub-projects.
Recognizing, of course, that resources are limited, and the primary purpose of the RFA is this 20,000 person assessment of genomic risk assessment and management. So that does need to be, hopefully it won't be monolithic, hopefully it will be a consensus process that everyone can come to agreement and have some important stake in terms of what is actually implemented. But when it comes to sub-projects, we will do our best to accommodate them. You should feel free to propose them, and we could even consider them after award as well. I'm not seeing any others. Are there any?
If you have any questions, please feel free to ask or.
Well, I did see one. Will a recording of this webinar be available after the meeting? If so, where and when?
Yeah, so I've sent the link on the chat box. I'll send it again. There will be a recording. The slides and the FAQs will be updated at this link. If you look at your chat box, the genome.gov, the eMERGE Genomics Risk Assessment and Management Network.
Hi, this is Karkulu. I had a question about the genetic testing. Will that be a high-density genotyping chip, or is that still to be decided? It could potentially be whole genome sequencing?
That's true to be.
Well, it's not going to be whole genome sequencing. I mean, there's just not the budget for that. But, you know, what exactly that will be, it needs to be the most cost-efficient, risk assessment that can be done, and recognizing that much of the risk assessment currently being done is not on rare genetic variants. It's on relatively common variants that can be obtained from a genotyping array. So one would expect that that's likely what it's going to be. But again, technology changes dramatically. Please propose what you feel is the best approach, or at least describe what you think would be the best approach. You don't have to include it in your proposal. And then we'll decide as a steering committee and as an institute what we can afford.
Thank you. Just follow up one quick question. So there was a mention of coverage for about $900 or $1,100 for potential testing. Is that limited to, is that everybody that has a high risk and might end up with those tests, or is it limited to just certain socioeconomic status patients?
That's for all individuals at high risk as determined by the assessment.
And that could be up to 25% of the cohort?
Correct.
Thank you.
We are expecting, however, that some of this, and an increasing proportion from the way things are going, will be paid for as patient care, as part of direct patient care, and that is increasing over time. These are not intended to be, extensive or intellectually stimulating investigations of the research value of various variants that are found in patients. This really is for clinical management and clinically indicated testing that just can't be paid for by a patient's insurance. That's what it's intended to cover. So it's going to be pretty limited.
Why is FDA approval required for the genotyping technology? So the FDA approval is because we're doing clinical implementation component of this, and so if we want to leverage what FDA has already signed off on for that effort.
In addition, we recognize that, and we love doing this, that genomic discovery is a wonderful thing, and it would be great to develop a new platform. When we've done that in programs like eMERGE, it has taken far more time than we had to be able to actually develop, validate, and implement them. So we need to get something that is essentially off the shelf, that is recognized by both CLIA-CAP and FDA. So that's why.
And there's a reminder for applicants to read the NIH grants policy statement section on patient care costs.
Mm-hmm. Oh, thank you, yes. And we should also note, for those who aren't familiar with the FDA and its role in genomic sequencing, they have flexed some regulatory muscles in regard to what gets returned to patients in terms of whether those are investigational devices and being used for research. That, again, is a process that they are fully entitled to implement, but we don't anticipate that if we're working with an FDA-approved or cleared genotyping array, that shouldn't be an issue. And so that's, again, another way that we're trying to get much closer to implementation of things that have been developed prior to the 5-year period of this grant.
Terry, Mark Williams here. Thanks for being proactive on the FDA piece. One of the problems with the FDA forces sort of a schizophrenic response, which is they approve the panel, but then they say you need to do confirmatory testing for certain things that are being returned. Has that aspect of this
yeah, sorry, Mark, you're breaking up. I don't know if you're standing over your microphone.
Could potentially, I'm talking, I don't know, does that help at all?
No, so we can't, could you either repeat your question or type it in, because we got every other word or so. Are you busily typing, Mark?
It was until you asked the question.
If you're on your computer, you might try calling in by phone, but it's maybe a little bit late. It is 5 minutes to 2. Okay. The other thing you could do is send the question in, Mark, because it sounded like, oh, you have it now?
I have, what is the rationale for disclosing PRS of pediatric population?
So for Mark's question, keep typing, Mark. Hopefully it'll be a short question. And if not, yeah, we see it here. Do you see it? All right, FDA has approved panels, but then required confirmatory testing for being returned. Has that been addressed? ... No, not yet. And so again, those are things that are going to have to be dealt with when we know what panels we're working with. Sorry, there was another question.
Oh, yeah. What is the rationale for disclosing polygenic risk scores to pediatric populations?
That would be we expect the investigators to make that rationale in their application.
Right, so there are polygenic risk scores and genomic risk assessments for pediatric conditions. So that might be one sort of thing that you might address providing back to children. There are obviously many more polygenic risk scores for conditions of adult onset, and as you're probably aware, the discussions about returning risk for adult onset, particularly late onset diseases, is a very active one, subject of great debate. And so that's one that we're going, again, to have to discuss within the steering committee. And it may be that some people will get some things returned and not others. Again, those are, sorry that this is poorly defined, but it's a rapidly moving area, and it's one that we want the very best applicants to propose how to address, and we'll bring it together into a consensus.
Next question, can sites focus recruitment on patients with a medical or family history that increases risk for one of the targeted phenotypes? Yes, but they'd have to explain how they would do that across the entire network.
Yeah, and why that's a reasonable strategy to propose. Again, we're not looking for high-risk people. We really are looking for sort of, a broad swath of the U.S. population.
We have another question. Is there a possibility that some patients would get genomic predictors and some would have these data held back as a control group?
So that we're not anticipating doing that. Yeah, I may have misspoken a little bit earlier, but yeah.
But you could use, I mean, there definitely can be a comparative group to make that assessment, but it's not expected that they would be included.
But what is anticipated is that whatever risk threshold we identify as being, above this level, you get risk information, that we would not provide that risk information below that level. And again, that needs to be developed with the steering committee. I think we need to.
We have 2 more minutes.
Okay.
If anyone has any last question.
Yes. What happens to the genotyping and phenotype, electronic phenotyping that we've done in eMERGE 1, 2, and 3? Is that being closed in eMERGE 4? There's no mention of it in the solicitation or RFA. And so I'm just wondering if that activity continues at all.
Yeah, I think we'll look back, John. I think that there actually is mention in the RFA of continuing the work that eMERGE has been doing in electronic phenotyping and linking to medical records, because it will continue as the eMERGE network. This is sort of a new emphasis of eMERGE, very much like the penetrance analyses were new emphases previously. We'll check that and get an FAQ out on that.
So we have one last question. So based on currently FDA-approved arrays, which would eMERGE anticipate using?
That is not a question that we at NHGRI are going to answer. That is one that we would like the applicants to propose what they feel would be the best approach. And if you don't feel that you can choose an array right now, you could describe what criteria of an array you would like to use for selecting it.
Okay.
Okay. Great. So our plan then, going forward?
Yeah, so it's 2 p.m. So based on the questions we've received today and any additional questions through email, we will update the FAQs on the website. There will be no further announcements to this update, so please check regularly. Just as a reminder, note that the webinar was recorded, and feel free to send any additional questions to emergerfa@mail.nih.gov. And thank you all.
And can you tell us the website, of course on the slide you can't see what the link is. You've sent the link out, but can you just tell us how we would Google that? Because that's how I find things on our website.
So you can easily just Google NHGRI eMERGE WebEx, or you can put in eMERGE Genomic Risk Assessment and Management Network, NHGRI, and you should get the link.
Great. And if you can't find it, just email us and we'll point you to it. Okay. All right. I think we're finished.
Okay.
All right.
Thank you.
Thank you.